I would like to welcome everyone to the Cohen Veterans Bioscience webinar series. This is a monthly program sponsored by Cohen Veterans Bioscience for brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit alliance whose mission is to transform research in veterans' mental health through translational research. We're focused on understanding the molecular underpinnings of diseases, such as PTSD and TBI, to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk you through some logistics. This program is being recorded and will be archived in the Cohen Veterans Bioscience YouTube channel and website. The presentation will be approximately 45 minutes in length to allow for a 15-minute Q&A afterwards. Please use the Q&A button to post your question. Today, our speaker is Dr. Patricia Kabitzke. Patricia Kabitzke is a behavioral neuroscientist who is working on preclinical models of neuropsychiatric and developmental diseases, such as PTSD, schizophrenia, and autism spectrum disorder. She has worked collaboratively with scientists across academia, biotech, big pharma, and nonprofit organizations, contributing expertise in studying design, assay development, analysis, and offering creative solutions to complex problems. Dr. Kabitzke trained in the Department of Psychiatry at Columbia University with Dr. Christoph Windermeyer and was a postdoctoral research fellow in developmental neuroscience and behavior with Dr. Peter Balsam. More recently, she spearheaded and managed the developmental disorders group at Psychogenics. Dr. Kabitzke is currently a senior program manager at Cohen Veterans Bioscience and an associate editor at the International Journal of Comparative Psychology. In this webinar, Dr. Kabitzke will speak on Animal Models 101, providing a general overview of animal models, the ways they are generated, and their contribution to scientific discovery. It is my pleasure to introduce Dr. Patricia Kabitzke.
Well, thank you very much for inviting me to present on this topic and also for the kind introduction. And thank you to everyone who is joining for the webinar. First, an overview of the topics that I will cover today. I'll talk about what animal models are, why they are a crucial part of the process of understanding and treating disease, and how they are generated. I'll talk about their massive contribution to scientific discovery, and also, importantly, some of the ethical considerations when using animals in research. Then I want to get a bit more specific and talk about model systems for post-traumatic stress disorder, including their current limitations and how Cohen Veterans Bioscience is working to overcome these limitations. First, what are animal models? One definition proposed by Dr. Joe Held, former director of the Division of Research Services at the National Institutes of Health, is the following. An animal model is a living organism in which normative biology or behavior can be studied, or in which a spontaneous or induced pathological process can be investigated, and in which the phenomenon in one or more respects resembles the same phenomenon in human or other species of animal. If we look closely at the first part of this definition, this hints at the general categories of animal models. How models are characterized overlaps with how they are generated, and we will get more into those details in the later section of the talk. The first are induced or experimental disease models. These are healthy animals in which a disease is brought about experimentally. They are the most common models used, and although they can be extremely useful to study an aspect of a disease, they rarely completely mimic the full manifestation of human disease in terms of its cause, course, and pathology. The second are spontaneous animal disease models. These model human disease because of naturally occurring genetic mutations. Again, spontaneous models often show similar symptoms and can have similar treatment options to a corresponding disease in humans, but do not necessarily have the same underlying causes. A very powerful example of this are spontaneous tumors in dogs, which occurs in 50% of dogs over the age of 10. These tumors share a variety of similar features with human cancer, and so this is a great example of how study and treatment could benefit actually both humans and animals. The third category are gene-modified disease models. These have been made possible because of rapid developments in genetic engineering and embryo manipulation technology over the last 15 years. These models are so important because they can get to the underlying genetic causes of many diseases. Most common are transgenic and knockout mice. In transgenic mice, new genetic information is inserted into the mouse genome, or by using techniques where genes already present are overexpressed. In knockout models, mice are engineered to lack the activity of one or more genes. For example, on the left is a knockout mouse that's a model for obesity, while on the right is a normal mouse. Genetic mutations can also be chemically induced. The fourth category are negative animal models. They are essentially control animals and are needed to validate an experimental result, and much care needs to be taken when selecting appropriate controls so that they are the same in every respect to the model except for the condition under investigation. The last category are orphan animal models. This is where a naturally occurring disease presents in a non-human species where there is no human analog. This could be because the disease is species-specific, or it just has not been described yet in humans. Why are animal models used in research? This relates to the second part of the definition, which describes a condition in one species that resembles a condition in another. How is this possible that discoveries made in one organism can provide insight into the workings of another very different organism? The reason we are able to use model organisms to research human disease has to do with early observations made by Charles Darwin and his proposal of common descent. Common descent describes how all currently living organisms on Earth share a common ancestor, and that over the course of evolution, biological pathways and genetic material were conserved across species. Some of Darwin's observations were of forelimb bones in vertebrates. The similar relational structure of these bones supported Darwin's theory of evolution and gave rise to the principle of homology, or measurable likeness, which is very important in animal modeling. In biology, structures and genes can show similarities in different species, and this is explained through descent with modification from a common ancestor across evolution. In the context of animal modeling, homology refers to animals that show similar causal factors, symptoms, and that can respond to similar treatment options as humans with that disease. This is the ultimate goal when developing an animal model, but they are quite rare. There are also isomorphic or partial animal models that show similar symptoms and can respond to similar treatment options, but that do not show similar causes of the disease. And predictive models that do not closely resemble the human disorder in any obvious way, but there is something about the model that allows the researcher to make predictions about the human disorder or its response to treatment. How are animal models generated? Some of the ways include genetics approaches, which can be done through selective breeding of animals with specific gene mutations or through genetic engineering. For example, one can use artificial selection when breeding animals to favor high and or low values of a particular trait, like good versus poor learners, or non-aggressive versus aggressive animals. This is a very familiar concept to dog breed enthusiasts and is used regularly when developing new rodent lines. Pharmacological approaches usually include inducing a disorder through the use of various drugs. For example, the administration of a neurotransmitter agonist or antagonist to reproduce human mental disorders, or the injection of a neurotoxin to parts of the brain as a model of Parkinson's disease. Environmental approaches include exposing animals to short-term or chronic stress or modifying their early life experience. For example, female rats show a range of maternal behaviors, from the optimal behaviors such as lots of licking or grooming of their pups, to their nursing posture to retrieving pups back to the nest when they wander off. The optimal nursing posture is where the dam hovers over her pups with her back arched so that she almost looks like a tent over her pups, and they all have good access to her nipples and stay warm underneath her without being crushed. Retrieval to the nest is also very important because pups do not thermoregulate very well early in life. Anyway, it's been found that these maternal behaviors cause changes in two molecular pathways of their pups. The pups of mothers that did not lick or groom them very much show high stress responsiveness and anxiety, and also do not consequently lick or groom their own pups. Conversely, the pups of mothers that licked and groomed them a lot later in life show less of a stress response, lower anxiety, and high licking or grooming of their own pups when they become mothers. And these effects are not entirely attributable to genes, since in cross-fostering studies, where offspring from these two types of mothers are switched, pups will show stress responsiveness and later maternal behaviors matching the mother that raised them, not their biological mother. How does this happen? Through epigenetic modification, which is a gene by environment interaction. The experience changes gene expression and behavior without changing genetic sequence. And epigenetic modifications can both be inherited and reversed. And by the way, researchers use the model we just went through as one of child abuse and neglect. A fourth way that animal models are generated, and I'll just cover these four today because they are the most relevant for the study of neuropsychiatric disorders such as PTSD, are disruptions induced electrically or within central nervous system specific lesions. Each of these methods has its strengths and weaknesses, and these techniques can be used together and may be useful to capture certain disease traits. Once a model is generated, how can we evaluate it? We can evaluate them based on different criteria. If they have face validity, this means that they show similar symptoms with humans with a disorder. Usually, though this is not very disease specific and very difficult to target with drugs, since the underlying biology is not clear or different between the animal and the human. If the model has construct validity, there is some underlying biology that can be measured and is in common with the human disorder. If the model has etiological validity, the disease process or progression between the animal and human are similar. And if a model has predictive validity, this means that we have the ability to predict how well a treatment will work in humans based on its performance within the model. Now let's pull back a little bit and talk about in what specific ways animal models have contributed to medical science. It is important to recognize that most of our progress was made possible because of animal studies. I'll just point out some highlights here. Before the 20th century, we understood the life cycle of the malaria parasite, developed early anesthetics, developed smallpox, typhoid, cholera, and plague vaccines. In the 1900s, corneal transplants and local anesthetics. In the 1910s, blood transfusions were made possible, and in the 1920s, insulin was discovered. In the 1930s, canine distemper, diphtheria, and tetanus vaccines and anticoagulants. In the 1940s, penicillin, kidney dialysis, open heart surgery techniques. In the 1950s, the polio vaccine, hip replacement surgery, pacemakers, and medicines for high blood pressure were developed. In the 1960s, heart transplants, MMR vaccine, and antidepressants and antipsychotics. In the 1970s, CT scans, chemotherapy, and inhaled asthma medications. In the 1980s, MRI, techniques for increased survival of premature babies, transplant rejection medicines, leprosy treatment. In the 1990s, combined therapy for HIV treatment was discovered, meningitis vaccine, and medicines for breast and prostate cancer were developed. And in the 21st century, we have deep brain stimulation techniques for Parkinson's disease, a cervical cancer vaccine, the bird flu vaccine, stem cells, gene therapy, and development towards a malaria vaccine. The same methods that have been developed to prevent and treat diseases in humans have also improved the lives of countless animals. Vaccines, antibiotics, anesthetics, surgical procedures, and other approaches developed in animals for a human are now commonly used in veterinary medicine. As a result, pets, livestock, animals in zoos live longer, more comfortable, and healthier lives. In many cases, treatments have developed specifically for animals. For example, rabies, canine parvovirus, distemper, and feline leukemia virus vaccines have kept many animals from contracting these fatal diseases. In this photo, an intradermal test for allergies is being performed in a dog. There are also now treatments for heartworm, which is a very painful disease and ultimately causes death in dogs. Animal research has also been integral to conservation efforts. Species that are endangered or have disappeared in the wild can now be managed or maintained through artificial insemination and embryo transfer. In addition, research on the sexual behavior of animals has made it possible to breed many species in captivity, enabling endangered species to be reintroduced into the wild. More knowledge of animals has also meant more humane animal control. For example, household and crop pests are often controlled using quite violent methods, but finding new methods based on aversive odors or predator models has been effective in many cases. In this picture, you see this owl structure, which is very effective in keeping birds away. Of course, there are important ethical considerations when using animals in research. This discussion often revolves around the relative value, or referred to as the moral value, of humans and animals. That is, when the needs of humans and animals are conflicting, which takes precedence. Interestingly, advances in biology and work with animals that began in the 1800s have provided some of the strongest arguments for the enhanced moral value of animals. By recognizing that the nervous system of all vertebrate animals are very similar, it's assumed that activities that will cause human pain or distress will likewise cause pain or distress in other animals. With this recognition, in addition to other discoveries such as tool-making and use, intelligence, advanced social behavior, and self-awareness in some higher-order species, the clear moral distinction between animals and humans has gotten fuzzier. To adapt to this increasing understanding of animals as a result of studying them, guides and regulations have been put into place and are constantly under review. A brief recent history. In 1963, an accreditation committee formed and published the Guide for the Care and Use of Laboratory Animals. Congress created the Animal Welfare Act, naming the USDA as the responsible agency in 1966. In 1971, the Animal Welfare Act was revised so that it required compliance adhering to the Guide, the Animal Welfare Act, and an additional set of principles for the use of laboratory animals. In 1979, the Public Health Service took over, requiring an animal care committee for each animal-using grantee institution and expanding the species covered to include all vertebrates. And in 1986, the term IACUC was formally introduced with an amendment to the Animal Welfare Act, and corresponding changes in Public Health Service policy and twice-yearly inspections of animal use facilities were mandated. What the IACUC is a committee required at all facilities that receives federal funds that must have a minimum of 3 members, including a chairperson, a veterinarian with training or experience with experimental animals, someone with no relation to the institution except for serving on the IACUC, a scientist with experience using experimental animals, and a non-scientist. What the IACUC does is that it reviews research protocols and conducts evaluations of the institution's animal care and use, and emphasizes minimizing the use overall of animals, ensures that animal use is performed in as humane a manner as possible, minimizing pain, distress, or discomfort, requires a veterinarian be employed at the institution so that the animals are looked after by someone trained in and sympathetic towards animals' needs, requires that analgesics, anesthetics, and sedatives be used where needed, requires that all personnel are trained and skilled, and specifies husbandry requirements. The IACUC performs inspections every 6 months and is required to report back to the Office of Laboratory Animal Welfare. IACUCs expect scientists to address the 3 R's, a set of principles meant to reduce the impact of research on animals. Reduction means minimizing the number of animals needed to perform an experiment. This can be accomplished by performing pilot studies to try to identify some of the potential problems in an experiment before a lot of animals are used, powering studies appropriately to find the number of animals required for robust, statistically significant results, and sharing information with other researchers so that experiments are not duplicated and we understand what results are robust and replicable. Refinement means refining experimental methods to minimize pain or distress wherever possible by using less invasive techniques, providing better medical care, and better living conditions, including environmental and social enrichment. Replacement means replacing experiments on animals with alternative techniques, such as experimenting on cell cultures instead of whole animals, using computer models, studying human volunteers, and using epidemiological studies. Without animal testing, the only options would be to end new drug development or to use human beings for all safety tests, which is likely unfeasible and unethical. Although animal experiments can't show that drugs are safe and effective in human beings, they can help decide whether a particular drug should be tested on people and eliminate some potential drugs as either ineffective or too dangerous to use in human beings. The value of animal experiments for predicting how well treatments will work in clinical trials is controversial, though, mainly because some of the treatments that look promising in animal models fail to translate to the clinic. This can partially be explained by flaws in how animal studies are sometimes performed, which can lead to systematic bias and results in bad data and making the wrong conclusions about how well a drug works. For example, if the treatment is not randomly assigned across the comparison groups, it is possible that animals that are thought to do particularly well or particularly badly are overrepresented in a particular treatment group. For example, sometimes the study is not balanced correctly so that all of the heaviest animals receive the treatment, and being heavier, they are also likely healthier, so at the end of the study, they could appear to be doing better than the non-treatment group, and an incorrect conclusion could be drawn that the treatment had a beneficial effect. The opposite case could also occur. Similarly, those who perform the experiment, collect data, and assess the outcome have to be unaware of the treatment to avoid bias. Well-powered sample sizes need to be calculated before the experiment begins to increase confidence in the findings and to also minimize the tendency to end the experiment once a significant effect is found. Preclinical results also sometimes fail to translate to the clinic because the results of an animal experiment didn't provide a correct basis for generalizations to the human condition. For example, when the disease under study is induced in animals that are young and otherwise healthy, whereas in patients the disease occurs mainly in elderly people with other chronic diseases. When the effect of a treatment is assessed in a very similar group of animals versus a group of patients that can show very different symptoms and causes. When only male or female animals are used, when the disease occurs in both male and female patients, when the model is not similar enough to the human condition, when delays to start of treatment in the preclinical experiments are unrealistic in the clinic, and doses are used that would be toxic or not tolerated in patients, and differences in outcome measures and the timing of the assessments between animal studies and clinical trials. Publication bias may also account for how positively treatments appear to work in animal studies that are then not seen in the clinic. That is, that neutral or negative animal studies are much more likely to remain unpublished, while neutral clinical trials are published. How does all of this relate to model systems for PTSD? Primarily, PTSD research is currently performed with mice and rats that are exposed to short-term, chronic, or multiple physical or social stressors, sometimes in conjunction with fear conditioning, and other times using fear conditioning alone in an attempt to induce behavioral similarities between the animals and patients suffering from PTSD. However, these models are mostly based on face validity. They're based on symptoms. They are not very specific to PTSD, meaning that they are also used to model aspects of other disorders, and it is currently not known if they have any construct validity or common underlying biology. Some genetic models are being developed in rodents based on associations found in clinical populations. These models may increase construct validity, but there are likely many genetic and environmental factors that cause PTSD, and they're likely not the same in all patients. In some, modeling PTSD is challenging in actually many of the same ways that modeling most neuropsychiatric diseases is challenging. The current animal models have significant limitations, including weak validation based mostly on face validity, and really no predictive power since there are currently no proof-positive drugs to treat PTSD. Individual symptoms observed in animal models likely do not have a simple, straightforward correspondence to human symptoms. For example, even while startle behavior in mice can be argued to correspond to startle in humans with PTSD, there is still an intellectual leap that must occur, since the related cognitive and emotional context cannot be determined in the animal. Also, sometimes there is just no analogy between symptoms in humans and what can be evaluated in mice, like intrusion or verbal memory deficits that occurs in PTSD patients. In humans, knowledge of the pathophysiology of PTSD is limited, and currently there are no objective diagnostic tests. Diagnoses are based solely on symptoms, signs, and course of illness, and how PTSD presents can vary in the patient population. Animal models are unlikely to mirror the full extent of PTSD. And lastly, the idea that tests are not models of a psychiatric illness. Many of the current models apply relatively short-term stress to normal rodents, which is very different from PTSD, where there is likely an underlying genetic vulnerability, which combines with random and long-term environmental stressors to produce long-lasting behavioral pathology. To address these challenges, Cohen Veterans Bioscience has created AMP It Up, the Alliance for Modeling PTSD using innovative technologies and uniform practices. Diagnosis of PTSD, again, is currently based on symptoms that can't be convincingly modeled in animals and are difficult to target with new drugs. So we will focus on the construct and etiological validity in the development of new model systems and in the validation of current model systems. Because PTSD can show very different symptoms and causes, we will seek to dimensionalize PTSD, modeling aspects of the disorder in animals. Increasing the translational validity of the models will get us to predictive validity so that we can get on to the business of developing treatments to relieve PTSD symptoms and perhaps preventing it. We will develop best practice standards for how studies of PTSD should be conducted. This will reduce bias and lead us to conclusions that can be trusted and built upon. This includes outlining statistical analysis plans to increase confidence in results and to make sure that every experiment gives us usable data. We will also explore innovative nano and imaging technologies to expand the kinds of tools that can be used in preclinical research. This includes improving experimental techniques and increasing capabilities to find biological measures in common with the human disorder. And we will investigate computational models to complement the limitations of animal models. For example, we could feed information into the model taken from what is known in PTSD patients and from our dimensionalizing approach of modeling aspects of the disorder in animals. This could be very useful to understand PTSD and in drug development. These are very exciting times, and we hope that you will come along with us on this journey. You can connect with us on our website, cohenveteransbioscience.org, or on social media where you can stay updated on our activities. I want to thank you very much for your attention, and I welcome your questions.
Well, I'd like to thank you, Patricia, for a wonderful and informative talk, and for everyone for watching and participating. If you'd like to go ahead and submit your questions, please use the Q&A button on the screen, and I can read those to Patricia. So we'll allow just a couple minutes for questions to come in. I'll just note again that this video will be archived on the Cohen Veterans Bioscience website, as well as our YouTube channel. So feel free to contact us at any time with questions, or Patricia with questions as well. I'm not seeing any questions at this time. Just give it a couple more seconds. So Patricia, I was just wondering,
yes,
from the perspective of developing a predictive model for PTSD, do we have any lessons learned from other psychiatric illnesses that could be applied to this specific disease?
Yes, absolutely. I mean, there have been a lot of trailblazers in this field. For example, the Centrix Initiative, which has done a lot of work in terms of schizophrenia work, and trying to understand constructs and the underlying mechanisms of schizophrenia to try to approach that disease in a more systematic manner, have given us great examples for how to approach these issues in PTSD.
Great, thank you. Well, there seem to be no other questions, so Patricia, thank you so much for your talk today. I'm sure there will be questions that arise in the future for you and for the AMP-It-Up program, and I just want to thank you again for this great talk. Thank you.
Oh, it's my pleasure. Thank you so much.